Now comes the sex determination, in which humans are of autosomes and sex chromosomes, in which humans have 46 chromosomes, that is in 23 pairs. Autosomes, it controls the somatic character, first pair and second pair. It controls the gender of human, that in 23rd pair it is male. Here the pairs of it is shown. Autosomes and sex chromosomes, allosomes. Autosomes are the chromosomes other than sex chromosomes. Number of autosomes in same male and female. Sex chromosome X and Y are the chromosomes which involve in sex determination. Hanking 1891 studied spermatogenesis in some insects and discovered chromosomal theory of inheritance, proposed by Walter Sullivan and Theodore Boveri in 1902. It states that the pairing and separation of a pair of chromosomes lead to segregation of a pair of a factors they carried. Certain united chromosomal regression with Mendelian principle and called it the chromosomal theory of inheritance. Important statements of chromosomal theory. Chromosomes are vehicles of heredity. They are transmitted from parents to offspring. That is, they are immortal. Two identical chromosomes with similar genes form a homologous pair. Homologous pairs segregate during gamete formation, that is in anaphase 1. Independent pairs segregate independently each other. Chromosomes are mutable. Genes are present on chromosome, hence they show similar behavior. Then comes segregation of homologous chromosomes. Here the diagram of it is shown. Then Morgan's experiment, which is Thomas and Morgan worked with fruit flies Drosophilus melanogaster to prove chromosomal theory of inheritance. Drosophila is a suitable material for genetic study because it breeds very quickly. Short generation time, life cycle 12 to 14 days. Breeding can be done throughout the year. Hundreds of progenies per making. They can grow on simple synthetic medium. Male and female flies are easily distinguished. It has many hereditary variations that can be seen with low per microscopes. This is male XY and female X. That male XY and female XX. Linkage and recombination. Linkage means physical association of two or more genes on a chromosome. Linked genes do not show independent assortment. Recombination means generation of non-parental gene combination. Here this is pachitin recombination. Then this is tightly linked. This is loosely linked. This is due to recombination. Morgan carried out several dihybrid crosses in Drosophila to study sex linked genes. Cross 1, yellow bodied, white eyes females. Cross 2, cross A and cross B. Morgan interested their F1 progeny. He found that the two genes did not segregate independently and the F2 ratio deviated from 9:3:1 ratio. Genes were located on the X chromosome. When two genes were situated on same combinations was such higher than the non-parental type. This is due to linkage. Then genes white and yellow were highly tightly linked and showed only 1.3%. Recombination white and miniature wing 39.2%. Recombination loosely linked. This was the third.